We start the session. Let me introduce the Tamil Nadu Science Forum. Tamil Nadu Science Forum is a voluntary science communication organization. It was founded in 1980 by few scientists in research scholars in Madras University who are doing physics, PhD in physics and in the Department of Nuclear Physics. They gathered and say that no, we do some research here. Why don't we go to the why don't we start an outreach program to talk popular science? They used to conduct lectures regularly on the university campus. People from different disciplines will come. Probably some people who are not connected to science will come. At some point in time, they decide to go beyond that and come out of the campus and go to the places, residential places, and talk about science. That has happened in the 80s. Having gained an enthusiasm because the response was good, then they think of going for a yatra. It started from Kanyakumari to Chennai. They go to different villages and organize lectures and demos. Things are happening. Then we get a lot of volunteers from different organizations, different districts, and the Science Forum was formed as a state level body. In the meantime, the government of Tamil Nadu has announced the program of literacy program. So TNSF was interested to take the literacy to the common man, and we get into the literacy program. Initially, we worked on the Pudukkottai district and Virudhunagar district, both declared as 100% literate district in the 1980s. TNSF was a full-fledged organization which was working on that thing. And we get a lot of volunteers in those two districts. And those district units become strong. Then the government was expanding the entire the literacy program to the entire state. Again, TNSF has become an organization in the forefront to carry forward this organization. Now, what happened after the literacy program? It becomes a state level body. We have a lot of the volunteers and members from throughout the state. And the question arises, are we going to stick on to only communication, science communication? Yes, that is an important thing. But apart from science communication, we need to get into other aspects of science as well. Then finally, the state level body has decided to split into 5-6 sections and work on those 6 areas. The first one is science communication. Then the second one, we bring science books, science magazines. We have started bringing a science magazine in 1987 onwards. It was a magazine in Tamil for children, Tulip. And 1991, we started publishing a magazine called Jantar Mantar, which is in English for college level student level. Then we also bring a lot of Tamil works in science. So that has happened in 19. That work was added. So we have a separate publishing group. We have a science communication group, science publication group. Another thing is we see that now we find that there is a gender inequality in science. So we have to work on that, and we form another group called Samam, where we want to bring more women into science. That is another group working group. And fourth one is we see a lot of environment and the developmental issues. Now, how are you going to trade between the science, the environment, and the development? When you go for more development, you are getting more damage to your environment. So we need to make aware of those issues. We have another group called the environment and development science. And finally, we have youth for science group. So there are 6 division verticals we are working on, different groups working from state level to the district level. And this group, the popular science group, in the science communication, we go to the schools, we conduct a lot of programs, particularly the Children's Science Congress. We want to motivate the students, children, to make sure that their analytical skill and analytical mind is improving. So we bring school teachers and they conduct a couple of days workshop to make students to become scientists, and we give topic. They taking the topic, select for a few students and make them to do the project, and we have a competition, and the students will present their project. Based on that, we select and award prizes. It starts from the school level, then we'll go to the district level, then the state level. Sometimes it was held at the national level, sometimes not, many times. It was stopped from last year onwards because central government has not stopped all those activities, science activities, but state government supporting us. And Children's Science Congress was one of the important thing. And Tolil Talent Test. These are all the things which we are working on the students, school level students. We thought that we did a lot of things for the school level students. What are you going to do for the college level students? Then comes the popular science lectures. The popular science lecture is always deal with the subject which is of current research. We had a lot of the lectures on the Nobel Prize in 2017 or 18 onwards. When the Nobel Prize was announced in October, first week of October, second, third, and fourth week we'll have Nobel Prize lecture on physics, chemistry, and biology, or medicine, or biology. So these three lectures every year we used to conduct, and we bring the importance of the work done by the Nobel laureate. Every year we do that. Apart from that, we have monthly lectures, and we also have several other activities. We have the workshop. So many things, so many activities are going on by the Tamil Nadu Science Forum. So with this, let me stop about introducing the PSL and Tamil Nadu Science Forum. Let me go into the topic. And today's lecture, I'm going to introduce the speaker. Yeah, Dr. Subhashri Deshikan will introduce the speaker. Just you only.
Everyone. Today we have a very distinguished speaker, Professor Mohanasankar Sivaprakasam. He's a professor in IIT Madras Electrical Engineering Department and leads the Healthcare Technology Innovation Center and Brain Center, which is going to be the topic of the talk today. After his US-based PhD and postdoc, he returned to India in 2008 to develop indigenous medical technologies, establishing the HTIC in 2011. HTIC and the Brain Center have spearheaded transformative healthcare innovations, creating 12 successful products impacting over 12 million patients globally, with 50 plus patents, 350 plus publications, and 350 crore rupees in funding. His efforts earned numerous prestigious awards. About the lecture today, the study explores advanced imaging techniques to map the entire human brain at cellular resolution. By integrating high-resolution microscopy and innovative data processing, it enables comprehensive visualization of neural structures, connectivity, and cellular organization. As you can imagine, it's done on fetal brains, as he will explain more to you. The findings offer transformative insights into brain function, disease mechanisms, and future neuroscientific research directions. That is for the introduction. I welcome you all to the talk once again, and I hope you enjoy it very much.
Thank you. So before we jump into the lecture, I want to make one more announcement. So we have a set of papers which has got QR code. So one such paper is to scan it, and your feedback, you can register your feedback, which will be helpful for us. So you please mention your mail and your mobile number so that we can communicate with you in future. Thank you.
This is fine?
Yeah.
Yeah, because I don't think they can hear this, right? So this is for them and that's for the web. Okay. Is this live or how does this work? YouTube live is going on. So is there a particular place I have to stand or how does this work?
There's a camera.
Huh.
Camera's there.
Yeah, but yeah, so that's what I'm asking. So I have to stay in a certain place. Okay. Glad, happy to be here. Some of you may not be aware that I'm actually an alumnus of Anna University, MIT campus. Nothing against CEG, but this is not. You missed that in the background, probably. That's relevant. So anyway, I'm sort of what I thought we will do today is we will go over the work that was done but I also want to spend enough time explaining to you how this kind of work happens, the how part, not just the what part. You know, these kind of very, very interdisciplinary work is something that's, you know, truly exciting, but it's also very challenging. And generally the students' feeling is, oh, I only know electrical engineering, I only know neuroscience, I only know biology. So, you know, how do I actually deal with other disciplines, right? In fact, you will realize science, even to some extent, quite a large extent, I would say, technology and engineering is actually becoming inherently interdisciplinary. So nothing like you can build a system with just knowing, you know, one discipline. You may not know all the disciplines, but you will have to work with people, right, who know this discipline. So that is something that we are going to emphasize, right? So I'll keep it a little light. Saturday morning, I don't think anybody wants to listen to the other person for more than 30 minutes, right? And after that, well, at least my students don't want to listen. After 40 minutes, they just go to sleep, right? So we'll do this, and then we will have a round of questions. So the headline, so to speak, is just a couple of months ago, IITM released the most detailed 3D high-resolution images of the human fetal brain, which some of you might have seen in the papers. And that's essentially the headline. Right? And we will sort of now look at how this was achieved, and we will start defining what is, you know, high resolution, right? And why is this challenging, and why is this scientifically relevant in terms of both advancing the knowledge and also creating new technologies to do this. Right? Okay, so that works. This center, as at IIT Madras, is about, physically it is 3 years old, I would say. Functionally, probably 4 years, 4.5 years. And it's a research center which is, you know, as I said, inherently interdisciplinary. It combines neuroscience, technology, engineering, and medicine, not just in its constituents of people, but even in the way it does the work. And it is supported by the government through the Office of Principal Scientific Advisor. And we're very fortunate to have Chris Gopalakrishnan, who is our alumnus, who gave the, you know, the initial seed support, took the risk, and continued to support it. We have Azim Premji's group, Premji Invest, and the Fortis Hospital Gates Foundation has been supporting us. And Prem Vatsa, who's another alumnus, distinguished alumnus of us, Saint Gobain, and the Prema Srinivasan Charities from the city itself. Now, what are we doing? We are imaging human brains at cellular resolution, and at a certain volume and speed, which is quite unprecedented. Right? So now let's go one level below and then sort of quantify and qualify all these statements. So today, the brain is actually the least understood organ, right, of, you know, vital organs, you would say. And it's been like this for a long time. Right? So generally, a field continuously moves, even if it moves slowly. I mean, there is usually some incremental movement every, you know, few years. But we have not seen the human brain at any detail better than a millimeter. Right? You can see the brain at MRI, through MRI. It's a millimeter. Millimeter resolution is not good. It's okay for diagnosis, to confirm diagnosis. Right? But you can't advance science when individual cells are at about 10 micron. What is 10 micron? Can somebody explain to me? What is 10 micron? How will you explain this to a high school student? No, that's arithmetic. Yeah, right. So I think you listened to my lecture already. So you're disqualified. Yeah. No, you're not cheating. You're disqualified. So it's about 1/5 or 1/10 of a human hair. The human hair is about 50 to 100 micron. Right? So it's a good way. So always remember, when you're doing numbers, you should have an intuitive way of explaining the number so that it's in your mind also, so that you get a sense of it. Right? So that is how much a cell. Right? There are different types of cells in the human brain. A typical cell is about 10 micron, 20 micron. In fetal brain, the cells are a little more smaller. Right? The smallest cell in the human body is a red blood cell, which is about 5 to 7 micron typically. So just to give you a sense of it. Now that's the first problem. And the human brain is unfortunately inside a skull. Like some of us may actually have a very thick skull, right? But most of us have a a very protective layer. So now there's another problem. So you have this incredibly complex organ inside a sort of an inaccessible place, right? Unlike an eye. That's why you look at, we can image the eye, the retina, we can image it at tens of microns today. Even not at scientific level, I'm talking about as a clinical level, right? You can go there and image the thing at around 10 to 100 micron. Right? That's amazing. The heart, interestingly, even though it is inside a rib cage, it's got an opening. Right? So that's why we are able to do the echo. So if the heart was actually inside a skull, let's just say, probably cardiology would not have developed to this level. Right? So it's very interesting how anatomy and of a certain kind advances and inhibits how much you actually know. And because the brain is inside a skull, you cannot get somewhere closer to it. Right? So you can only do radiological, which is CT or MR. Right? Now you'll be surprised, the images of the brain that we have actually are few decades old. That's all. There has been hardly any advancement. Yeah, there is some digital gimmickry that you may do, right? Saying, oh, I'm going to zoom in, I'll do some image processing and everything. But fundamentally, we have not seen the whole human brain at anything better than a millimeter. And this is a long-standing problem. Now, we are talking only about the structure. We are not even talking about the function. And in biology, generally, right, form determines function, meaning structure determines function. So if you want to understand live human brains, how these billions of cells, how many cells do you think will be there in the human brain? Take a guess. Billions, actually tens of billions. Right? We actually don't know exactly. Some people say 100 billion, some people say 120 billion. There is some reports 80 billion. So we don't even know. This is number of neurons. I'm talking about only neurons. Then there are glia, there are other support cells, then there is vasculature, then there is connectivity. Connectivity is usually 10 to 20 times of the number of neurons. So we're talking about trillions of connections. Right? Now, how are we going to advance something unless you get some reasonable view of all this? I mean, that's usually how you advance anything. So you advance the what part, then the how and the why part follow. But if I can't even know the place, right? I mean, then how will I build anything there? How will I analyze something there? So this is the fundamental, is one of the unsolved scientific problems, right? Like the way the space is. Right? About 30 years ago, people used to say the space, the gene and the brain. Right? The gene is sort of, at least sequenced. We've still not understood what all affects the gene, right? The expressions and the factors affecting the expression. But, we have a handle on it. Right? The space, it continues. But the brain, actually, we have not made any advancement. You'll be very surprised. And this has implications on the clinical, which is medical applications. So I'm sure many of you have, had an encounter with a neurologist. Actually, it's quite, very sobering and somewhat depressing to think about it. Because we don't understand the brain enough. There are hardly any tools, be it drugs or, surgeries are used to remove tumors, but we can't go and fix something. We can't rewire it like the way we are doing valve replacement, right? The eye, we can actually go and fix few things there. We can't do that in the brain because we don't have even a basic, a good map of the human brain. Right? So because the science is not advanced, we don't understand the human brain well enough, our medical advancements have not happened. So that's why you look at Alzheimer's, Parkinson's, any neurodegeneration, which basically when you age, the brain ages, and unfortunately all of us are outliving our brains. Right? This is becoming a problem now, which used to be starting a Western problem, but now I'm sure many of you, even the youngsters, you will notice it. Right? Your grandparents are now getting older and sort of losing their mind. And that is now becoming more and more common. We are seeing that. And my generation and following that, your generation will be the largest old generation in the world. We are going to, right? And many of us, by the time we reach there, our life expectancy will be touching 80. Average life expectancy. Already our life expectancy is sort of getting close to 70s. So this has another implication. So unless we have some handle on how we will deal with this, it becomes a huge societal problem. So it has an implication in terms of socioeconomics, has an implication in policy. Right? And with the declining population, when you have an aging population, which also is neurodegeneration, and this is now playing out in many places in the world, in Europe, to some extent in US, in Japan, right? You're starting to see that. So this is the larger picture and understanding of why we need to understand the human brain more. Now, before I tell you how we do it, because there are youngsters here, I want you to think about, now if I want to image the human brain at cellular resolution, what can I do? Or what should I do? It's a thought exercise, right? So you don't have to worry about the feasibility of technology. That for a moment, let's just say somebody will do it for you. How will you do it? I'm talking about the whole brain. You can take pieces of the brain, right? Which is what is done in biopsy. You take pieces of the brain, you look under a microscope. Right? But here we are talking about 1300 cc, about few 100 billion cells. Right? So you obviously cannot take pieces out. Right? Which means the only way to do this is you have to image the whole human brain after extracting it. Right? There's no other way. Which is actually, how most anatomy is developed, right? If any clinicians here, you probably have not realized that, anatomy and physiology to some extent, that's now become, people try to skip those classes, right? But actually the reality is medicine is today because, we understood the anatomy and physiology quite well. Right? So the only way to look at the brain at this level of detail is, remember, any radiological thing cannot do better than a millimeter. And we are talking about, if you have to look at a cell at 10 micron, you have to image it at least 1/5 to 1/10 of it. Only then you can, else you'll only see a dot, right? If you image it at 10 micron. If you have to see the structure, you'll have to image it at least, 1 or 2 micron, right? Half a micron is what we are doing. Now, light cannot penetrate. So you should actually ask, is there a way I can actually penetrate light inside the skull? So obviously not. Well, not only a light cannot penetrate the skull, even if I take out a tissue, light cannot penetrate beyond few hundred micron. Actually, few hundred micron is already pushing it. Right? 100 micron is what I can penetrate, which means I have no other option than to slice the brain. Right? You have to slice the brain at very, very thin sections and then image it, the entire brain, and then put it back together. Right? So this is a hard problem. Right? So you can imagine at every level, how will you acquire a brain? It's soft. Well, even before it becomes soft. So let's say, how will you acquire a brain? So you cannot acquire a brain, 3 days after death, it would have been decomposed. Right? So you need to acquire this brain while it is still, the cellular architecture is still there. Of course, it's lost its functionality, but we are looking at the anatomy, not the physiology of the brain. Right? So how do you acquire this in less than 8 to 12 hours? Right? At a very high quality manner, both high quality in terms of its process and high quality in terms of ethics and documentation, everything. Right? Then now you have a brain. How will you now slice it at 10 to 20 micron? Why 10 to 20 micron? Because we want to see each cell, right? So you don't want to cut it at 200 micron. Then there are close to 20 cellular layers to it. How will you do it? And if you have ever cut something very thin in your kitchen, you will realize the thinner you cut, easier it breaks, right? Physics. Right? So how will you cut something very thin, reliably, and across the entire brain? So if you do a quick calculation, you will realize if it's an adult brain, we're talking about close to 10,000 sections. Right? If it's a fetal brain, somewhere around 2,000 to 4,000 sections. How will you cut this? Then how will you handle it? Okay, it's cut. Now it's there. Now you can't just pick it up just like that, right? So you will have to find a good instrumentation that can pick it up. It can, some of you understand histology and histopathology, you have to be able to stain it with particular dyes. Right? Then you have to image it microscopically. And this is large scale imaging. So generally microscopy will do imaging of few millimeter by few millimeter. We're talking about few, well, actually almost half a feet by, a feet, less than half, right? 15 centimeter by 10 centimeter, a little more actually, 20 centimeter, right? Almost 2/3 of a feet. How will you do such large brain sectioning? Imaging. Plus, you do all this, you will now produce huge amounts of data. Right? So you can do the quick calculation. If I'm doing half a micron per pixel imaging, we are talking about 150 gigapixels of one human brain section, one section. And that's around, let's say 1 byte per pixel. So you're talking about 150 gigabytes, right? Times 10,000. Right? Do all the math. We're talking about petabytes of data. Right? So petabyte is a large data. Even in today's world, right? Huge amount of data. Right? It's comparable to, let's say, Google Maps. Right? The entire Google Maps will be in several petabytes. So this is what we went after about 4-5 years ago, actually 5 years. And today, so now one thing which you probably have realized, what is a neuroscience problem essentially is a technology gap. Okay. Neuroscientists cannot solve this problem by themselves. You need the engineering and the technology disciplines to build this platform. Right? And you need the clinicians, the doctors and the medical researchers to work with you to acquire these brains in a certain way and interpret the images and the data in a certain way. And by the way, we are not only doing the cellular level imaging, we do the MRI also, because what is our ultimate goal? We want to be able to, in future, if I have the MRI of the live patient, I should be able to extract the cellular level image. And for that, I need to have done enough MRI plus detailed cellular level of enough brains. Then I can build these models. Right? So that's what we are after. Now you're probably wondering, like, man, this introduction and this is not making any sense. So this is this electrical engineering professor. What is he doing with this? Right? So there is some background to it, and I'll briefly mention the background, and that's somewhat relevant to the point I was making. Is this work is new, about 4-5 years, but our group has been developing medical technologies in collaboration with doctors and various subspecialties of medicine, as well as clinical sciences, for over about 13 years. Right? So some of you might have heard of HTIC, Healthcare Technology Innovation Center. It is an IIT Madras research park. Till date, it has developed close to 12 products. It has developed various technologies. 12 products today commercially available, which some of you might have even used it without realizing it. Until date, it has reached close to 14 million patients in various fields: ophthalmology, endoscopy, patient monitoring, diagnostics, robotic surgery, and so on. So out of that is actually this work actually really came out. Right? And it really happened when Mr. Chris Gopalakrishnan, our alumnus, in around 2015-16, he started to have a conversation with us saying, can we do something at the interface of neuroscience and engineering? Right? So the inspiration actually starts from there. Right? And he said, I think this area is quite wide open, even at a global level. So it's not that India is too behind, because the area itself is very wide open. Can we do something significant? So we looked around. There are multiple ideas, multiple projects that we thought about. And in around 2018, we decided to pursue this project, saying that we will do whole human brain mapping at cellular resolution. And to give you a context, at that time, the mouse brain was what was being mapped. Mouse brain is about 1 centimeter cube, right? 1 by 1 by 1 cc. And monkey brains, which are about 30 to 100 cc, they were getting mapped. So the human brain, even if it's the smallest brain, fetal brain, second trimester, you're talking about 100 cc. It's an adult brain, you're talking about 1300, 1200 cc. So that's 1000 times of the mouse brain. So it's a truly challenging problem. And essentially, you need technology at scale. Right? Only then you can crack this. You cannot say that, this will work for, these days for this many sections of this part of the brain. The rest of the thing I don't know. Right? Then you cannot do it at scale. So that's how the project started. And 2020, it's 5 years. Last week was actually perhaps the 5 years since that project was officially sanctioned by the PSA office. And you're probably wondering, in 2020, something happened. Some of you are very young. Right? The project started and then COVID came in 2 weeks. So we couldn't even start the project. So it's very interesting, right, how there are non-scientific challenges that you have to overcome, and that's actually part of the pursuing large science, big science projects. We overcame all that, and the entire technology platform was actually built during the COVID period in 2 years. And in fact, the center was inaugurated when COVID got over. So 2022. And since then, we have been getting support from various people. And today, this center is, producing truly, first-in-class results, which are shared globally. Right? It's not just, hidden in IIT Madras. It is put out globally so that other people can see it, they can comment on it. And we collaborated with close to 20 globally leading researchers in various aspects of neuroscience. And this center, sometime when you're not too far from here, you'll be able to visit. It has close to 150 people of various kinds, neuroscientists, engineers, different types of engineers, a lot of computational people, and large number of doctors who collaborate with us. So I don't know how much you can see from there. It's a little unfair for the people who are at the last row. So this is the process which, the pointer doesn't work. This is the process which, takes a human brain. The acquisition of the human brain itself is actually a challenging process, right? After death, there is a series of paperwork that is done with consent from multiple people, and then the brain is acquired, and it's also perfused because the brain has blood in it, right? And we want to clear the blood. There are different ways to clear the blood. One is, you soak the entire thing and then hope it diffuses away, but that is actually not very effective because, as you know, the brain is a series of, it's basically the blood is in the tubular structure, right? The vasculature. It's a very, very fine vasculature. So what we do is we created a process of perfusing the brain. Right? Using the carotid and the vertebral artery, you actually perfuse, you create a fluidic circuit, and then you perfuse the entire brain so that it's cleared of blood. Right? Then it is put in a cryoprotection solution. Then it is put in the fixative. Right? then you are now ready to freeze the brain, right? And if you're wondering why we should freeze the brain, if you want to cut something at 10 micron, 20 micron, right, you're not going to be able to do it without making it really hard, solid, beyond solid actually, right? Absolutely strong. So you freeze the brain at minus 80 degrees Celsius, then the harder problem comes, right? So typically cryosectioning, right? Some of you, how many of you have heard the word cryostat? 3, 4 people. No, you're disqualified again. 3 people, right? So cryostat is basically, is used for large animal experiments, right? So where you freeze the tissue and then you use a certain mechanism, machinery to cut through the tissue and create sections of, several tens of microns, right? Now, that can be done for smaller volumes, smaller regions, but you're talking about 1000 cc and with a area of 10 by 15 centimeter. Very, very challenging, right? Try thinking of, cutting a very large bread at, 10 micron. It's obviously going to break somewhere, right, in the middle or towards the end of it. Even if you cut it, how will you transfer this into a glass slide so that you can start doing the section? So we have developed a series of very sophisticated instruments to do all this, right? It's a little difficult for me to explain this, especially for people in the back. So I will sort of give you the qualitative picture of it. We section this and then we transfer this very, very thin tissue sections into glass slides, and then we stain them with various immunohistochemistry markers. It's basically think of different colored dyes, right, for approximation. Then we image them. And remember, when you're doing microscopy imaging, we want to get all this work done, I mean, in our lifetime, right? And it cannot go on for tens of years. So we have to develop microscopy systems that can do this reasonably fast. And this is classic light microscopy. They can do fluorescence, they can do ISH also if needed. But this is classic light microscopy. This is not electron microscopy. In fact, even this is petabytes of data. So you no point in doing electron microscopy of human brain of this volume. It's not possible. So today we can actually, image an entire human brain section in less than half an hour, right? One human brain section. So 10,000, if you multiply all that, today we have a throughput of, we can do an entire human brain in less than a month. Right? And this is one of the fastest in the world today, right? And we continue to accelerate it. And this is something I referred to in the early part of my talk, which is, why are we doing this? If you reflect on this, and this is a comment by Francis Crick, who won the Nobel Prize in 1960s for his discovery with Watson, the double helix DNA structure. And the statement was made about 30 years ago, 30 years after he won the Nobel Prize, 30 years before today. And, what he says is quite stunning, right? He says, unless I see what is in there, unless I get the structure of the human brain, I can't interpret the activity of living human brains, right? Naturally. And today we can do this, which is at 1 millimeter, right? That's all. And you can see there's not much you can actually get from there, right? I mean, for clinical things to confirm a certain bleed, certain lesion in a certain region, tumors, that's okay. But if I really want to understand the brain, you've got to get to some level of micron, which is not possible. So you have to create these reference maps. That's how, medicine actually advances. So you create these advancements in medical sciences, and then you convert them into tools that can be used in live patients at a certain cost and a certain speed, in a certain area and a form factor. That's how MRI came, and that's how, everything, whatever you looked at, is all driven by some scientific advancement, which was eventually translated into medicine. Right? So that's what we are after. And to give you a global context, practically everybody who has done this work in brain usually start with small animals, mouse, then they moved into medium-sized animals, monkeys, right? Then they moved to humans. So we somehow took a big risk in 2020 that we'll start with human brains directly, right? So today it has worked, and of course, I can probably talk about it. It could have also not worked. So there is always a risk, and without that risk taking, leap is not possible. If you have to leap, you should be prepared to fall, right? And usually the risk is quite high, right? And that is something that you should all remember. These kind of science projects have a big risk, right? And if you ask me, I would say this risk was probably higher than 50-60%. It's a huge risk if you think about it, because literally all of this has to work, and has to work at a certain scale for this to happen. And today we are, we are at a position where we probably will be able to create the largest repository of brain maps in the world, right? And we are after it. And this is how the brains look like. Just to give you a sense of size, that is slightly disproportionate because of the screen. I think it's squished a little bit. So starting from 17-week fetal brain. And why are fetal brains important? You probably have already figured out. That's where all the clues of how the brain itself develops is there. Which structure, which region was formed when, and then the how part. If you want to answer, then you've got to go and look at the fetal brains, right? And these fetal brains are usually intrauterine deaths, where the fetuses don't survive. This is not stillborn or medical terminations. These are intrauterine deaths, usually happens in early part of the pregnancies. And these are extracted. And for adults, usually the causes of deaths vary, like natural causes, infections, road traffic accidents, right? Mostly accidents. And old age is natural causes, stroke, various infections, right? So that's how we acquire these brains. And obviously you can't see the gigapixel here, but you can actually see them when you go to the viewer. And that is the kind of resolution at which we can see the brain. And now comes the hardest part. Right? Now, how do you put it all back together? Because we wanted the whole human brain, right? That was our goal. We want to see the whole human brain at cellular resolution. And this is a computationally very, very challenging task, right? Because you've cut the brain. Now the brain is in thousands of sections. Now you've got to put it back together at an accuracy of few microns. Right? And we worked with NVIDIA on this, and today, this information, this data is actually quite public, right? You can actually look at it. And not only have we, this doesn't seem to play, it plays, right? Not only have we imaged them, we have annotated. So that is essentially, this is map making, right? I can take satellite pictures of, Chennai. Not only have we done that, we have annotated different regions, right? Different land masses, water bodies, and the pathways. And that is a very, very important part, because once you have that, that's how you create these reference maps, right? And what we have today, it is actually, in this public now, it is the most advanced second trimester human brain data set that's out there with very, very detailed annotation, right? And like this, we intend to do this across from first trimester all the way to 100-year-old, right? And that is what is being done as we speak. And I should say published already. In fact, this week it is published, not just accepted. It is been published in the oldest neuroscience journal in the world. And this is today the largest publicly available data set of human fetal brain. And now, you're probably wondering, like, okay, now how do I make sense out of all this data? One is qualitatively, I can see it, which region and everything. So this is another big challenge. Right? So we are building a computing platform which can do all kinds of analysis, all kinds you can imagine. How many cells are there in what region? What is the density? What are the different cell types in which region? How does this brain compare to a brain with a disease? What is the density of a certain types of cells in a certain type of region in a certain disease? Right? These are the questions that both the neuroscientists and the doctors want to know. Right? And so that is being done as we are speaking. And in fact, we are one of the first world to have counted all the brain cells. Right? First time. Right? And that is actually currently under a review for a publication. And we're coming towards the end of the talk. And you probably already realized that this work cannot be done just by one discipline or one, you can do it in one place, but you really need different kinds of discipline, right, to come together. And that is how, large science projects, I would say large technology projects get done, right? And that is something that we have been fortunate. And we have a very strong global collaboration network, people from different parts of the world. And this is what, in one way I can summarize it. We are creating this very, very integrated, multi-layer, multi-scale brain maps, right? Which is from MRI to cellular to various immunohistochemistry, which essentially is a chemical map, you can say. And we are doing molecular mapping of certain regions of the brain, because it's very hard to do for the entire brain. It's not only very expensive, it's very it's practically there are some limitations. With all this, our ultimate goal, the translation is, we want to advance disease diagnosis and treatment of brain diseases, which is sort of stuck today. So, this is the vision of the center. Whatever work we do, we want it to be of globally high quality, and we want it to be globally relevant and globally available. So I will sort of stop there and say that some really exciting work is happening right here in the city, not too far from here. And this is putting India on the global map in this frontier area of brain science. Thank you very much. How far it is? We just started. So medical science has not yet gone. Not yet. So for example, we are currently doing this for diseased brains. Okay, yeah, that's it. So right. Can have some. Yeah. So understand. Yeah. So for you to do this, you need to have what is called normal, which is neurotypical, and the diseased. Right? Then you do the comparison, then you ask the medical question, then the clinical researchers will look at it from, oh, this patient had, then you go one level deeper, right? In terms of, one level away, I should say. You look at the kind of treatment protocols that are available. Is there anything we can do? So if you find a certain cell type in certain region, in a certain way, which you did not know that it was there, that gives you a certain clue. Right? Are there any other brain centers like this across here? In India, this type of work, we are the only one. So remember, this type of work also requires multiple disciplines to come together. Right? It's a tall order to aim for something that's big, and you should be able to. The institution should support that kind of large projects. Yeah. Talking about the implants, they must be having some database, right? No, actually, no. You'll be very surprised. Even the deep brain stimulator, which has been going on for now 30, 35 years already, we don't have a very detailed understanding of how the actual stimulation works. We know it works to a certain level, which is actually true. I mean, many cases in medicine, you will relate to this. Carefully, but let's just wait for the.
Really brain image. Your all over entire brain image influencing any changes in the psychotic patients.
So for that, we have to actually get enough brains of those patients. Only then we will be able to compare.
இது வந்து நம்ம பேசிக்கா வச்சுக்கலாமா சார் ஸ்டாண்டர்டைஸ் பண்ணலாம்.
ஆமா. ஸோ இப்போ வந்து நம்ம வேற வேற ஏஜ் குரூப்ல வேற வேற டிசீசஸோட ஸ்டாண்டர்ட் ரெஃபரன்சஸ் கிரியேட் பண்ணா அதை வச்சுட்டு டிசீஸ் பிரெயின் வருதுன்னா இதை கம்பேர் பண்ண முடியும். இந்த ரீஜன்ல இது எப்படி இருக்கு அப்படின்னு. ஸோ ஒரு பேசிக் ரெஃபரன்ஸ் கிரியேட் பண்ணோம் மற்ற ஏஜ் குரூப் எல்லா ஏஜ் குரூப்பும். இப்போ அந்த வேலை தான் ஸ்டார்ட் பண்ணிட்டு இருக்கோம். அதுதான் பண்ணிட்டு இருக்கோம் இப்போ. அதுதான் போயிட்டு இருக்கு. Through MRI.
Okay, but how do you take it to the molecular?
So that's exactly how science advances, right? So you create enough data sets of invasive brain imaging, which is what it is. And that's why I explained to you, even here you can see, we take the MRI also of this brain. So you create MRI and cellular level of the postmortem brain, and you create enough of this data, right? So that in live patients, when you get the MRI, you will be able to derive the cellular level. I mean, not AI means modeling. I mean, what AI is being thrown around. Yeah, everything is AI now, unfortunately. So I'm very careful in saying yes or no. People are doing even arithmetic using AI. So
Direction to my house. Showing
yeah.
Sir, on what basis the storage you are showing up? On what basis TV 300 TV adult brain? On what basis the
You
storage?
can calculate, no? If it is 1 pixel, is 1 byte. You can calculate.
So it is limited, you are saying.
What does it mean? I'm not following the question.
So, I mean, the storage of I mean, TB in the sense, I thought it's a storage memory you are talking.
Yes, it is.
Okay. So, so you are able to say that we are limited with 300 TB of storage in adult brain?
No. Sorry. That's not. I probably came in a little late.
Oh,
so
yeah.
This
Sorry, I
is
missed.
yeah. So this is microscope. Once you image this data,
Okay.
how much is this data itself?
Okay.
I'm not talking about storage in the human brain.
Oh, okay. One general question, sir. Like, I'd like to know about the like three different types of ideas. Like, dreams are there, simulation is there, reality is there. Like, how do you differentiate in a? I mean, using a. I mean, what do you take off? Like, how the brain sees things, like dreams or.
So that is still far away. So
it differentiates.
yeah. So we don't know, right? So that is, if you want to see how the live brain today is behaving, you do fMRI. Right? fMRI is functional MRI, as we call it. Right? So you can only say this brain region is activated. That's all we can say. And we're talking about this region, meaning we're talking about several millimeters to centimeters. That's all we know at this point of time. How does it happen? Which signaling pathway is triggering this? That we don't know, because we actually don't have the structure. So I mean, you take Chennai, it's not an exact analogy. You take the city, right? Unless you know exact fine details, then I can't reroute traffic, right? Right? I mean, you get the picture. For you to understand something and be able to manipulate something, you really need the details of it. So this is, we are very, very far if you look at. That's why the brain is one of the ultimate frontiers, right? Even not just in biology and medicine, but even as a science. It's such a, and you can You can just wonder about, how complex it is and how little energy it's taking, right? How do we learn? I mean, I've not even gone to that level. How do we learn? How do we store? I mean, you asked how much brain, I don't know how much, but I can remember so many scenes of so many movies, but people can even enact it so well. So that means that I can store this. How does storage work in the brain also? We don't know. We actually don't know anything. Right?
The storage area. They would tell.
Sorry.
It's dynamically
Yeah.
keep the
I think you have to wrap it.
Thank you for that interesting talk. I have a question about the mechanics of doing this,
Okay.
about the slicing very precisely. So how much time does it take to make one slice? And then I have a
It
follow-up.
takes about 2-3 seconds to make a slice. In fact, you actually have to make it fast. If you make it very slow, no,
It'll collapse.
it'll collapse. Exactly. So, I mean, the short answer is it'll collapse because it's now flapping, no? This guy is hanging at 10 micron, 20 micron. So you better do it fast and transfer this guy. It's frozen, but 10 micron is very thin regardless. Doesn't matter how frozen. I mean, the human hair, right, is actually quite strong if you look at it. I mean, how much can you hold it? It'll warp. It'll also bend in its own weight. That's the other challenge because it's pretty big, no? 10 centimeter by 20 centimeter at 20 micron. Yeah, using different stains. Exactly. Different histochemistry வச்சு different cell types பண்றோம். அது பண்ணிட்டு இருக்கோம். You have some
the diseased brain.
No, I didn't say that.
Accidental cases, good brain also.
Of course. There's nothing called a good brain or bad brain.
Not disease.
So not known disease. So typically, yeah. So when it's a clinically documented disease case, that is one. So the neurotypical, right? What we call neurotypical are usually, no documented disease, but some other cause of non-neurological related, right? You can have a heart attack. Of course, heart attack can sometimes have a direct effect on the brain. That depends, right? It depends on the type of heart attack you have. Right? The heart attack not always translate into a significant neurological condition, like that. So typically, you can say neurotypical and diseased. Right? Same with neurotypical fetal brains and developmental disorder fetal brains. So some developmental disorders are, there even at a very early stage, very, very clear, very, very early stage. So like this, we have acquired close to around 250, 260 brains.
So basically now this work shows the neural mapping of the brain.
The cellular level mapping,
Yes,
not the connectivity yet.
Yeah, cellular mapping. And to bring it to as this question of clinical application, so you're going to overlap this with the MRI data and
That's
then
the first step.
Right, okay.
First step, because that's the proximal. If you want to translate something, that's probably the closest you can look at because there's already MRI there. Right? So if we can have an MRI and we can have the cellular level detail next to it, you'll be able to think of it as a co-pilot. Since we talked about AI, that's the closest analogy I can
So
give.
now still we have MRI of 1 mm. So you're going to have many sections overlapping with this from the cellular mapping.
Yes.
I'm sure MRI technology is going to, we do CBCT now, cone beam computer, which is like 0.2 millimeter we count.
Few hundred micron you can get to, but cellular level is too far. No?
Yeah,
It's about 1 micron. So there are fundamental physics limitations why you cannot push radiology imaging beyond this. And remember, I mean, this is what you're talking about is, acute situation of, that particular area. But for example, you take Alzheimer's, there is no understanding.
Yeah, that's right.
There is absolutely no understanding. Even the tau protein hypothesis is, it is a hypothesis and you get exactly opposite results. So how does Alzheimer's, what is Alzheimer's itself is actually still not well characterized. And neurodegeneration is a global, it's not just a point related thing. Right? So got to have a sense of how does his brain look when it is diseased and where all it is affected compared to a neurotypical brain. That's the first step actually.
So how about instead of MRI going to a positron emission tomography scan, which will be more better than MRI, do you think?
Yeah, but it cannot get better than few hundred, I mean, a millimeter and then
Yeah.
1/3 of a millimeter is all you will get.
Yeah. But instead of going for MRI overlap, I'm asking about whether PET overlap will be
Whether
better.
we can do PET. See, one challenge in when you do this, we are doing DTI, right, for the connectivity. One challenge when doing all this is your logistics. Now you've taken the brain out, right? How much access will you get in certain imaging facilities limits what all you can do. That's a short answer.
So because I'm interested in more of craniofacial diseases, so we have always a neuro problem attached with the craniofacial problem. So looking forward for more information.
In fact, one of the things that we are looking at is, as I was telling you, we're interested in looking at the cranial nerves, right, starting from the ganglion, if not the nerve fully, but that has a lot of information, the brain stem and the cranial nerve and the ganglion. Right. We are looking at it.
Yeah, thank you.
Thank you.
So extending from what he was asking, your work is basically looking at the structure, isn't it? You already have MRI, but you're taking MRI to a very, very 10x level. How do you think this will sort of give information to the function of the brain? Brain is a very functional.
Sure.
We do have a lot of information on the structure of the brain.
In fact, the only
Where do you see the correlation? How do you correlate with the function? For example, say in Alzheimer's or dementia, structure like brain tumors, you can see it, you know what it is. But I'm talking for functionality of the brain, which is what the problem is, which we don't know much about.
Yeah.
How does your research help in there?
So as I was saying, form determines function, mostly in biology. Right? Because there is adaptability that happens over a period of time, but the form meaning structure, determines function. This is one of the axioms of biology. Now, today, the way we can look at brain function is through patient describing it, or EEG, or fMRI. These are the only options you have. You can look at certain regions of the brain by ECOG, which is you put some electrodes, not deep inside, but at the dura level. So there are, even today, regardless of whether you advance this or not, there are not many ways to actually look at the function of the brain, except at gross level. Gross meaning we're talking about fMRI, right? EEG, but EEG is used more for looking at specific patterns, mostly for epilepsy, right? So the function, because it's determined by form, only if you get the details of the pathways. So the function in one way is determined by the different cell types, because somebody asked, and the connectivity between them, the pathways. So if you want to get to function at that level, which you're talking about, this is the first step. Because today there is no other way to do function. EEG is usually mostly EEG is used for epilepsy and sleep studies. No, we actually have to first determine which structures in Parkinsonians and Alzheimer's patients, how are they? What are they? Which cell type? For example, I was saying Alzheimer's, even today it is actually not clear which region is affected, which proteinopathy as we call it, right, is affected. It's actually very loose hypothesis. I mean, for decades now. Right? I mean, before even we go to the signaling pathways, we're talking about yes. If you get a Parkinsonian brain, right? We have a couple of Alzheimer's brain, Parkinson's brain, we still don't where to get one. That's the first thing. You actually do immunohistochemistry and figure out which receptors, which type of cell types. And sometimes we do cell immunohistochemistry markers of even very, very large number, like as high as 20, 30, right? Then you create, the different colors which you saw in the fetal brain, they were actually more of density. But if I can do the coding as different cell types, right? So which are the cell types? How are they in Parkinsonian patient? That's the first step. So if I ask you to find differences, you need to first have the two maps in front of you. That itself is missing now today. And that's why you find there's hardly any advancements in drugs in Parkinson's and Alzheimer's. In fact, not many, most pharmaceutical companies are not working on anything, because you need some advancements so that you can actually take a bet and then say, okay, I will now do a control experiment. If you don't even know what is your baseline, how will you do control experiments? I think we should wrap it.
Here is that 100 plus score of human brain. But during the answer, you said 250.
250 brains we already have. Okay. We want to get to 100 plus human brains in terms of getting them imaged.
These are all stored in IITM now? These brains?
Some of them, and some of them are with our medical partners. Because once you extract the brain, you can store them. That's not the challenge. Okay. Okay. Okay.
But it will hit out of the what happened?
Sure. Saturday morning.
People want to see it later. They want to record it. Anyway, is there any other questions?
Last question.
Last question.
I heard the project long before the Connectomics, which is also similar of yours, I believe. And like, how is it different from yours? I
don't know what you have done, so I mean, I cannot.
Connectomics is an international project on the case of they talk on the cells. Connectomics project is there. They've been running for a long time. It's an international project. That's all I know, and I heard it a long time back. They also did the mapping as you do
At small regions.
Not small regions. They talk about the entire mapping, I believe. And
no, the entire map of the human brain, if it's there available, then it would be out by now.
No, they talk in the informatics part of it.
I don't know. I can't answer that because I don't know what you're talking about. So I cannot refer to it. I think.
So actually, my question is, are you going to the research on the disorders based பண்ணி போக இல்ல trauma based பண்ணியும் எப்படி சேஞ்ச் நடக்குது அப்படிங்கறத மைக்ரோஸ்கோபிக்கலி பார்க்க போறீங்களா இல்ல ஆஃப்டர் trauma disorders கிரியேட் ஆகுது ஃபார் எக்ஸாம்பிள் எம்ஆர்ஐ எடுக்கும்போது சிலருக்கு வந்து பாத்தீங்கன்னா trauma நடந்திருக்கும் பட் when you are taking
நான் சொல்றேன் உங்களுக்கு trauma is only one particular type of disorder, right? So we are looking at diseases and disorders. There is a very small difference between disease and disorders. We are looking at different types, so that's why I said 35 categories of age and diseases. So we're looking at trauma, which is well documented, right? And different diseases, which are not necessarily trauma as you define it. Stroke, neuroinfections. We're looking at the rabies brain, right? Because we still don't know. In fact, somebody was asking. So we still don't know how rabies affects the brain. We know it affects, but we don't know how it affects. In fact, it actually travels along the axon, but without killing the cells. It's quite a mystery, actually. Okay. So we are looking at different diseases and neurotypical, both.
Also, I'd like to ask, are you people concentrating on traditional systems also? Like in Siddha, Ayurveda, there is Burma and Madras. So if that attack is there, we can't.
That's a good question, right? So exactly. So if you want to study all of that, you need to have a certain map, right? Because right now only the patient is responding, right?
Mapping,
you need the map for you to actually say, oh, this is the pathway. So you take the brainstem pathway. We're actually doing the brainstem work as we are speaking, right? So once we get all the nuclei, then the next step of doing the pathway, right? And even if you don't do the connectivity at axon level, which is very hard to do because axons are few microns, right? Even if you get gross pathway at few millimeter level and few centimeter level along the brain, then you will know which brain regions are connected to which other brain regions in what form. Okay.
Extension வந்துட்டு பாத்தீங்கன்னா இப்போ வந்து பாத்தீங்கன்னா நிறைய டைப் ஆஃப் things like how I love care because it does there like that in Chinese also chronic in weekly like that things are there. இது வந்து treatment aspect in the application
Of course, that's what I'm saying. So if you get enough brains of enough diseased patients and typical patients, then you have the documentation, right? Then you do what you call clinical research in one way, right? So you say, this particular patient, we had this finding, but we didn't realize that this area. So that's why you need a lot of clinical research. After this work is done, right? There is neuroscience research, which is advancing neuroanatomy as a field by itself. You need the clinical thing to overlay that information, the information which you said, and fill the missing pieces. And also some gaps will always be there. You make that, you fill that gap using other knowledge, right? And that is how you advance. So that's a lot of work.
This is starting, and this is not going to be never ending. It's going to keep on going.
It is.
Until all the diseases are.
So today, if you look at the people say, after a long time, there is a certain critical mass of brain-related research happening in the world. Because it's not been done for a long time. So even in US, the so-called brain initiative started only in 2013. Until then, it was funded as like individual projects. A critical large force. It started only now. So in one way, we are, that way when Chris Gopalakrishnan said, this is a wide open space, but India is not too far behind. Because, other people have also not advanced anything too significantly.
This is my last question.
No, I don't have a problem. They will.
One thing, so a couple of months back, we had a lecture from Srinivas has given a lecture. He's working on the brain. He wrote a book on that. His approach is totally different from, instead of focusing on the structure, you focus on the function. So how are you going to connect all those things to develop a function of a particular thing? So what is the difference between the thing which you are focusing on?
Yeah, so what happens is, this actually has happened in physics, right? Where you do the experiments, right? At a physical level, you glean knowledge, you build models, and then there are purely theoretical conceptualization based modeling that happens, and then they reconcile, right? One will actually, because when you do these experiments, eventually you will want to simplify this knowledge into a certain models and certain theory. Hopefully the theory, the word theory in biology is very rarely you actually get a very strong theory in biology. It's not that easy to actually develop theories. But that's how it happens. The field gets advanced because when the field is so wide open and there are so many gaps, right? And certain people will be doing things at only molecular level. Yeah. They'll be like, I can't do whole human brain. I will take these cell types and I will go really deep and look at how the RNA is expressing itself. Right? So you will have different kinds of knowledges being developed.
Yeah, there are two kind of school of thought. We Ramachandran school of thought. He focus only on the function, not on the things. His lecture is starting with, we have a style in Marina Beach, we have 3 billion sand. What is the problem? Is it complex? He says it is not complex. Let us focus on function rather than structure.
Yeah, because
that's why his approach was there.
Yeah. So as I said, because you have 3 billion sand, that's why it's a beach.
Yeah.
Right? So you also cannot say that 3 billion is nothing.
Yes.
So I mean, it's very easy to see. The problem with analogies and metaphors is it oversimplifies and approximates to a point. It can be severely misleading. So you need the detail. At the same time, you need the simplification, because if you don't have the simplification, I mean, how else are you going to, translate this into practice?
Correct.
Right? You can, a patient walks in, you cannot say, sir, this particular cell, he'll be like, man, thala valikudhu enakku marundu kudunga. Right? That's all he will want to know, right? You cannot say this cell in this region is happening this way. Let me show you the detail. That that you can do it with your lab, right? So both are needed, but without the detail, you will not get to this level of drugs don't work this way. I think we should wrap it. It's been a while.
So time is the only constraint. This video is available on YouTube. You can watch it again. And while giving the feedback, if any further questions are there, you can share with us. We will try to get an answer. So